Good day, students. I am Professor JP and I'm going to discuss to you the plant and animal cell. So, at the end of the lesson, students should be able to give the description of cells, differentiate between plant and animal cells, identify the functions of each parts. But first let us define what is a cell. Cell is the smallest and most basic functional unit of life that holds all of the biological equipment necessary to keep an organism alive. Plants and animal cells are both eukaryotic. This means that they share many similarities, but they also have some differences. First, in the cell shape, plant cells have usually square or rectangular shape thanks to their cell wall. Animal cells do not have a cell wall, so they have irregular and flexible shape. For cell wall, plants have a top cell wall made up of cellulose. Only plant cells have this structure. Animal cells do not have. For cell membrane, it is also called plasma membrane, which is found in both types of cells. It controls what goes in and out of the cell. For membrane-bound organelles, first we have nucleus. This is an important organelle found in both plants and animal cells, which operates as the control center where all genetic material is stored and managed. For plant cells, it is found mostly in the periphery of the cell, while in the animal cells, it is found mostly in the middle. For plant cells, it is found mostly in the side or the tucked away at the edge of the cell because of the large central vacuole of the plant cells. For cytoplasm, every plant and animal cell has this segment. Its job is to support and suspend organelles and cellular molecule to maintain cell integrity. For endoplasmic reticulum, we have two types, the rough ER and smooth ER. The rough ER has a structure called ribosomes, while smooth ER lacks ribosomes. Rough ER are concerned with protein synthesis, while smooth ER functions for lipid synthesis and detoxification processes. Ribosome produce proteins as directed by the cell's genetic code. Both plant and animal cells contain these. They are also called as protein makers. It is also the site for protein synthesis. For mitochondria, which is also called the powerhouse of the cell, as we all know, which generate most of the cell supply of the adenosine triphosphate or ATP, which is required for chemical energy sources in a cell. The mitochondrial compartment of plant has less in number than that of animal cells due to differences in their physiology. Moreover, animal cells, particularly the muscle cells, require more energy demands, hence they are more numerous in animal cells. Golgi body, also called Golgi complex or Golgi apparatus, is found in both plant and animal cell. They serve a role in modifying, sorting, and packaging proteins and lipids for secretion and delivery to various parts of the cell. For plant cells, Golgi bodies are more numerous, but animals have fewer but more complex Golgi bodies. Next is the centrioles. Centrioles are not found in most higher plants. They are generally absent except for cycads, ginkgo, bryophytes, seedless vascular plants, and charophytes. On the other hand, it is present mostly in animals because it is vital in cell division, which facilitates the organization of the spindle fibers that separates the chromosomes during mitosis and meiosis. Lysosomes. In animal cells, a tiny sacs called lysosomes are commonly found. They are not typically found in plant cells because plant cells have a structure called the lytic vacuole, which is structurally different from the lysosomes. Lysosome contains hydrolytic enzyme, which is necessary for breaking down waste material and cellular debris. Next is the vacuoles. In plants, we have a large central vacuole which maintains turgor pressure, store nutrients, and isolate harmful materials for plants. On the other hand, for animals, it is much more smaller and less in number. It functions in storage, osmoregulation, and aid in endocytosis and exocytosis processes. Next is the plastids, which are found only in plants. Plastids are present and are involved in functions like pigment synthesis, storage, and photosynthesis. In animals, it is generally absent. Next is the locomotion structure, specifically cilia, which is generally absent in plants. In animals, they typically exhibit ciliated cell structures on their apical surfaces which serve various functions such as for mobility, for feeding, and sensory perception. Next is the mode of nutrition. In plants, they are known as the main food creators. Why? Because they create their own food through a process called photosynthesis. Hence, they are called as autotrophic organisms. On the other hand, animal cells rely on others for food because they don't have chloroplast, which is involved in photosynthesis. Hence, they are called as heterotrophic. For a summary of the table, we have here the cell parts and the comparison between plants and animal cell. So here is the summary for cell shape, for cell wall, which is only present in plants but not in animal cell. They both have cell membrane. They also have nucleus but found in periphery for plant cell, in middle portion for animal cells. Both also have cytoplasm, ribosomes, and mitochondria. For Golgi body, they both have these, but more prominent in plant cells, but fewer and more complex in animal cell. For centrioles, they are generally absent in higher plants but are present and typically found for animal cells. For lysosomes, are generally absent in plant cells but found in animal cells. For vacuoles, we have large central vacuole for plants, smaller and less in number in animal cell. Plastids are only found in plant cell, while ciliated structures are typically exhibited by animal cells but can be also found in lower taxa for animals. For mode of nutrition, plant cells are considered as autotrophic, while animal cells are considered as heterotrophic. To conclude this topic, plant cells and animal cells have shared features and characteristics as they are both under the eukaryotic classification, as well as they also have differences that make them unique from each other, such as their shape, the presence of cell wall and cell membrane, differences in membrane-bound organelles, locomotion structures, and their mode of nutrition, which stems from their differences in anatomy and physiology. Such differences and structures are tailored to their function in their respective environment and their role in ecosystem. That's all. Thank you.